Everyone, welcome to Bioacademic YouTube channel, and on today's video we'll be talking about one of the most amazing process that happens inside my body, your body, in all of our body every single second, and this process is called DNA replication. Now, I know that sounds a bit technical, but don't worry, by the end of this video you will understand exactly how DNA replication works step by step in simple language. That is what we are here for, biology made simple. Now, without further ado, let's go to now. What is DNA replication? Now, before we dive into DNA replication, let's get familiar with the word DNA. Now, DNA stands for deoxyribonucleic acid, and what that means is that DNA is the molecule that carries genetic information in every living organisms. DNA is responsible for carrying genetic information in me, in you, in plants, in animal, in birds, etc. You can think of DNA as your body's instruction manual. It contains all the codes that tell your cells what to do, how to grow, how to repair themselves, and even how to look. DNA is found in the nucleus of our cells. That's like the control room for each cell. And if you could zoom in on it on a microscope, you will see that DNA looks like a twisted ladder of what scientists call a double helix. Each rung of the ladder is made up of chemical pairs called bases. We have adenine denoted by the letter A, we have thymine denoted by the letter T, we have cytosine denoted by letter C, and we have guanine. Now, these bases always pair in a very specific way. A pairs with T and C always pair with G. That is the rule that keeps your genetic code stable. Now, for example, it's like having a secret code where A always makes T and C always means G. If you ever break the code, the message gets messed up, and that is how mutation can help. Do you understand what I'm trying to say? Okay, let me put that again. I said it's like having a secret code where A means T and C always means G. Like, let's say you and I are communicating with a secret code now, and we don't want anybody to know. And in this secret code, hey, if I say A, it actually represents T, and if I say C, it actually represents G. Now, if by any chance now one of us decides to break the code, the message definitely will get messed up, and we won't even understand ourselves anymore, because we already agreed that A always means T and C always means G. But when you're not saying B, that it represents T, or you're saying Y represents C, now it will be so messed up that even you and I won't be able to communicate effectively the way we used to communicate again. That is exactly what is happening. That's exactly what DNA is doing in our body. So if this DNA is not doing its work properly, that is how mutation gets to happen, and we know what mutation is. Now, what is DNA replication? In case you don't know what mutation is, don't worry. As we go further, we'll be talking about mutation in our other videos. And anytime I make that video, I think I will drop a link attached to this video so that you'll be able to go to that video and be able to watch it. Now, what is DNA replication finally? DNA replication is the process by which your cell makes an extra copy of its DNA before it divides. When I say extra copy, I mean, when I say exact copy, I mean like photocopy. Like when you're photocopying, let's say you're photocopying a note. You know, you writing a note and you photocopying a note is different thing. When you're writing a note, you might make a mistake, or your handwriting can be different. Even though you are the one that wrote the first note, your handwriting may be different. You might make a mistake. You might decide to like make some part lengthy. You might decide to make some parts shorter. But now, no, you are making a photocopy. It has to be exactly the way it is in the original note. Now, that is exactly what we're talking about in DNA replication. Like I said earlier, I said DNA replication is a process by which your cell makes an extra copy of its DNA before it is divided. Remember, DNA is found in the nucleus of the cell, so the cell now is responsible for making an exact copy of DNA before cell division occurs. You see, every time a cell divides, whether it's to grow new tissue, heal a wound, or make reproductive cells, it needs to pass on the exact same genetic information to the new cells. I'm very sure you all remember mitosis and meiosis. In case you don't know that, don't worry. As we go, we'll talk about this also. So DNA replication ensures that every new cell that has the same instruction manual as the parent cell. For example, think of it like copying a recipe. Before you share a recipe with someone else, you make sure every ingredient is written correctly so that they can cook the same dish. Right? Do you understand? Let me go again. Let's say a friend of yours made a nice pot of soup, and they be, oh my friend, I love this your food tastes very very nice, and I love it and I want it. Now all you have to, and your friend be like, oh yes, I can share my recipe with you, and you make sure every ingredient is written correctly so that when you are cooking you can get the same result. Do you understand? That's exactly what DNA replication is talking about. DNA replication is basically a biological process by which a cell produces two identical DNA molecules from one original molecule before it divides. Now, what are the main enzymes involved in DNA replication? When we talk about enzymes are like the workers in a biological factory. Each one has a specific job that helps the replication process go smoothly. Here are the main enzymes and what they do. Now we have number one is the helicase. Helicase is like the zip in your clothes, but in this case, it doesn't just act like a zip. It's just like an unzipper. The holy work of the helicase now is to unzip your clothes. It doesn't zip it. So, like I said earlier, I said DNA is a double helix structure. So initially it's a double helix structure. So helicase now, it breaks the hydrogen bond holding the two DNA strands together. You know the hydrogen bond have like A is got adenosine is connected to thymine, while guanine is connected to cytosine. So this helicase will now break up the hydrogen bond holding the two DNA strands together. This opened up the double helix into two single strands. Now after it opened, it becomes two single double strand. Now from this image on the screen, I'm very sure you can see this structure of the DNA, which is the double helix structure, and you can see in between these two strands, you can see that they are both, from the first one, I think the thymine is represented by this purple color, and this adenine is identified by this green color. You can see how they are both bound together, how they both hold the two strands together. Now the work of this helicase now is to unwind, to break down this bond holding these two DNA strands together to become two single strand, to become two single strand. This one strand will be at one side, another strand will be at the other side. Now then we have primase. Primase is the maker. It adds a small piece of RNA called a primer, showing where DNA polymerase should start copying. Now after this double helix structure has unwind, the primase, now we had a small piece of ribonucleic acid called a primer, showing where DNA polymerase should start copying. Now imagine it like drawing a starting line before a race begins. Do you understand? Before you start a race, where all the racers, we have to stand at the starting point. This is where you're going to start the what's it called, the race. That's exactly what the work of primase. Now moving on, we have DNA polymerase. DNA polymerase is the builder. It reads the original DNA strand and had matching nucleotide to form the new strand. Now you already know that this double helix structure, they divide, their helicase comes and break down this bond and they become single strand. When they become single strand, you know this nucleotide, this adenine, they breaks. This for them, this number one, this first one now, this thymine, we go to one strand. This adenine will go to one strand. Now DNA polymerase will now read the original DNA strand and add matching nucleotide to form the new strand. So let's say this one here now, the one with thymine, is a single strand. Now this DNA polymerase will be the one to read this. Oh, this is thymine. So since this is thymine, then the matching nucleotide will be what? Adenine, to form this new strand. It's like typing out a duplicate of a document line by line. Down number 4, we have ligase. Ligase is like the glue. It seals small gap between the new formed DNA fragment, making the strand complete. Now, you know, when this thiamine, this strand with thiamine, when it is like a single strand like this, and this DNA polymerase reads an original reads the original DNA, be like, okay, this is thiamine, then to make it complete, we need to add what's it called, matching nucleotide, which is adenine here. Now this ligase now will be the one now to glue, we had to act as a glue to glue this new thiamine and new adenine together, making the strand complete. It's like using glue to fuse the seam of a paper you cut and joined back together. Now we'll now talk about how this DNA replication happens step by step. The first thing is the DNA unwinds. That is the initial stage. This is like what I just explained, but now in a more in-depth form. Now, step 1, the DNA will first unzip. This is the initial stage. The process begins with the enzyme helicase attached to the DNA molecule. Now, the helicase, which is a protein, which is an enzyme, and you all know what enzymes are catalysts, you know. So now it attaches itself to the DNA molecule and it starts to unwind and unzip the double helix by breaking the weak hydrogen bond between the base pairs A with T and C with G. Now this unzipping creates a Y-shaped structure. Can you see it? Can you see it? Can you see it? Called the replication fork, where the DNA strands are separated and ready to be copied. Now step 2, the primer is now added. Once the DNA is unwound and is open, the cell needs to mark where copying should begin, right? Primase lays down a short piece of RNA, which is ribonucleic acid, known as primer. The primer tests DNA polymerase. Hey, this is where you start adding new bases. Do you understand? This is where you start adding new bases. For example, imagine you're photocopying a long book and you place a sticky note on the exact page where you want to start copying. That is what Primase does. For example, let's say you want to copy your friend note word for word, and you're not copying that exact all the notes. So you'll be like, okay, this is exactly where I'm starting from, and this is exactly where I'll be ending. So you need to indicate, okay, yeah, so this is where this note I'm writing, this is exactly where I'm starting this note from. Do you understand? That is the work of primase. Now step 3, the new strands are built. This is called the elongation stage. Next, the main builder DNA polymerase jumps into action. Each separated DNA strand now serves as a template. What do I mean? You know, we all know templates. What template? Like a sample, yeah, like a template. Now DNA polymerase read each base and add its complementary base. What do you mean complementary base? What? Let's say the first strand now is adenine. Then this polymerase will now be like, okay, yes, since you're adenine, then you pair with thiamine. Do you get? That is the work of DNA polymerase. However, there is a twist. The two DNA strands runs in opposite direction. Because of that, the leading strand is copied smoothly and continuously, and the lagging strand is copied in a short section called the Okazaki fragment, since it's run in opposite direction. Do you understand what I'm trying to say? Now let me go again. I said there is a twist. These two DNA strands runs in opposite direction. Because of that, like this new DNA that is added, they both run in opposite direction. The leading strand is copied smoothly and continuously, and the lagging strand is copied in a short section called the Okazaki fragment, since it runs in the opposite direction. From here you can see the leading strand, and you can see the lagging strand. You can see this arrow. You can see this arrow, and you can see this is continuous. Yes, and you can see this continuous. You can see this the part where we labeled Okazaki fragment. For example, it is like building two roads at once. One road is straight and easy to pave, but the other has curved so it's difficult in a small connected part. Now step 4, joining the fragments. Now that the new DNA pieces are formed, there are small gaps between the Okazaki fragments on the ligase strand and where the ligase comes in. Ligase act like a glue, like I said earlier. It seals the small gaps and join the fragments together, making one smooth, continuous DNA strand. Example, imagine glue together puzzle pieces to form one complete picture. That is exactly what ligase does. Now step 5, proofreading and error correction. So before the process ends, DNA polymerase double check it works. It proofreads the new DNA strand from any mistakes. If a base was added incorrectly, it should remove it and replace it with the right one. This step ensures that the new DNA copy is a perfect match to the original. For example, it's like when you are done writing your essay, you proofread the original essay to say, okay, yeah, this spelling is wrong. No, let me remove this sentence. Yeah, this makes sense more. Do you understand? To check to make sure everything is correct. Now, the final step is the restore stage, where the two identical DNA molecules are finally formed. Now, finally, the process is complete. Now the cell has two identical DNA molecules, each containing one old strand and one new strand. This is called semi-conservative replication. What do you mean by semi-conservative replication? Because half of the original DNA is conserved or kept in each of the new copy. For example, think of it like making a copy of recipe. You keep one original page and one freshly written copy, but both say exactly the same thing. So, in summary, DNA replication is like a high precision copy machine that works inside every living cell. It unzips the original DNA, builds